Okay, let me get back to it. Okay, so bacterial genomics is something that I worked on during my thesis, a PhD thesis. Right now I'm working on the multi-omics data analysis, particularly working on the bacteria and the host interaction with various data analysis, which involves single-cell RNA-seq, metabolomics, and microbiome. Um, yeah, so this brief introduction about me. And so the main goal of this workshop is for you guys to perform the entire process of bacterial genomics, starting from genome assembly and genome-based identification and annotation, and then finally with the comparative analysis. Um, Bear in mind the technical details will be explained kind of in brief manner, because I want everyone to follow up with the command, with the pipeline and command line that I'm going to provide. Yeah, but the if you're interested for further details about the algorithms about each program, there will be a reference provided at the very bottom of each page. And for those of you who are taking it for credits, there will be a quiz and assignment after day 3 of this workshop. So the due date, you're gonna get about a week to finish them. So due date is May 7th, which is next Friday, I believe. And so for the outline for the workshop, since today's day 1, I'm gonna provide a general introduction, and we're gonna mostly spend the time on setting up the environment, and then we're gonna start assembling a genome from the raw Illumina FASTQ files. And in day 2, so this is gonna take a few hours, I think, to finish up. So we're gonna put it on the command line and wait until it finishes off. So there's gonna be day 1 material. For day 2, once you have the assembled genome, you're gonna measure the quality of the assembled genome. We're gonna learn how to identify uh, the species level um, with both 16S rRNA, which is a marker gene for species delineation, and using a whole genome. And then we're gonna learn how to infer a phylogenetic relationship. And in day 3, we're gonna focus more on stuff like the genome annotation, the functional stuff, genome annotation, the concept of core and pangenome, how to perform comparative genomics analysis, and then visualize some of the stuff that we generated. So most of the pipelines and softwares will be run through the Hoffman 2, which is a cluster hosting system provided by UCLA. Um, so it consists of large computing resources, um, which is connected by the network. So a user can either access each of these nodes interactively. So what's important is that this control node, the master node, which sends the commands to each of these nodes. So user get so this is the shared computing resource. So depending on how busy they are, you may get the node or you may not. But hopefully, each one of you can get a node for this workshop. To connect Hoffman 2, you need a SSH client. If you're using Mac, you can just use the terminal or PowerShell for Windows, but I prefer to use PuTTY because it's kind of old school method. Yeah. So how many of you are using a Windows? Can you guys give me a thumbs up if you guys are using Windows? None of you? One, two. Okay, so I guess rest of you are using Macs, right? Can you give me a clap for the Mac users? Okay, majority of you. Okay, then in that case, you guys can use the terminal. I think that's easier. But for the Windows user, you guys all have this slide, right, from the Google Drive. So you guys can click this link, and you can download the portable version of the PuTTY, which is 64-bit. Yeah, so once you have it, you can turn it on. And on default settings, you can type in the address for the Hoffman 2 server. So hoffman2.idre, kind of confused. IDRE UCLA, okay. UCLA.edu, and you can click on open. It's gonna ask your ID, Hoffman 2 ID. Just gonna type mine in. All right. So once you see your ID at login 2, then this means you're connected. For Mac users, sorry about that. For Mac users, you guys can open up your terminal by clicking the launchpad. Click on others, and there's going to be a terminal there. Click on that. Do you guys see my the VMware right now? Because it's showing me a warning sign not to share this application. Do you guys see it or not? Give me a thumbs up. If you... Oh, okay.
We only see your slides.
Okay, okay. I guess I'm not allowed to share the VMware screen then. Okay. That's bummer.
Can you is it a window that's open?
Yeah, yeah.
You can unshare
I'm
your.
sharing my entire.
It won't let you
I'm
share the
sharing
slide.
my entire screen right now.
Oh, really?
Yeah.
All right. All right. Yeah.
Do you guys see it now?
Yeah. I see the top part says VMware.
Okay, okay, cool. Okay.
It says player, but no.
Yeah, but you guys see the Mac screen though, right? No?
Yes, yeah.
Okay. Thank you. Okay, so you can click on the launchpad and click on others and click terminal. And then you can type in SSH and then your Hoffman 2 ID and then the address Hoffman 2 RDRE UCLA.edu. And it's probably gonna ask you to type in your password. So you guys go ahead and yeah. So once you got connected, let's try to get an interactive session. Let's try to grab a node, available node in the Hoffman 2. So you can type in this command. Probably not this one. This one is for the CPU power. But if you ask for the CPU power more than one, it's gonna take a while to grab a node, I think. So let's type in this command and grab a node, I guess. So qrsh dot dash l h underbar rt equals, this is for the time. So how much time you're gonna use the node. So we're gonna use it for 6 hours. And h data, which means the RAM, the memory you're gonna use. We need, I think we need at least 8 gigs of the RAM. So once you type that, press enter, and it's gonna take a few moments to get connected. And same for putty, you guys. You guys can type in the same. Okay. And next thing we're gonna do is we're gonna set up SFTP module application, which is uh, for the file sharing with the Hoffman 2 server. So you can click on this link as well. Yeah, so for the Windows user, the website will automatically detect your operating system, and it's gonna lead you to the Windows program. So you can click on download and download the free version. And you guys can click on and get it installed. I already have it installed on my computer, so I'm not gonna install it. So anyway, yeah. So for the Mac again, um, I guess you can go to the same web page. Sorry for the delay here. Yeah. What? Oh yeah. So you guys can access to this web page. Uh, and again, the website will automatically detect your operating system, and it's gonna ask you to download the client. You guys can click download. And then once you download it, you're gonna have it file name under the tar and VZ2. This is the compressed version. You guys can extract it just by double clicking it. Yeah, so once you extracted the compressed file, you're gonna see this little icon here. So you guys can go ahead and start opening up. And it's gonna send you the warning sign, but just, you can just ignore it. Just get it open. All right. And let me get back to my Windows screen. Yeah, so you guys will gonna see probably something like this. So you can click on the button on the top left corner here, click on it, and trying to make a new site, name it after Ubuntu, and change the protocol to SFTP. And then for the data transfer, there's a specific address that you have to connect to. I mean, this provides the faster file transfer speed. So DTN dot Hoffman2 dot IDRE UCLA dot edu. Leave the port blank. This one's the default value is 22, so leave it blank. So logon time, I'll set it to ask for password and type in your user account and press connect. And it's probably gonna ask you the password and type in your password. Yeah, so you will see the list of folders that is under your home directory of the Hoffman 2 server. You can simply drag and drop files to this part of the window. Um, so is everyone following right now so far? Or am I going too fast? Okay. And let's get back to Putty. Let's see if we have if we got any nodes here. Yeah, so on the Mac one, I got one node right now. It says node 2191. So I got connected. Same for my Windows version here. So I'm gonna work on mostly on Windows side since my computer is on Windows. Next, you need to download the data. So let's go ahead and make a directory for our workshop. Get rid of it. So let's make a directory, make dir qcb workshop, and let's try to download some data from. Yeah, it can take some times actually. Okay, okay. You guys didn't type in this command, no, right? Did you guys also type in the PE shared option here, or okay? So I guess you guys just need to wait there. So what you can do is just open up the separate terminal for now. Um, you can click on new window and get a separate connection. I think you're allowed to download the file on the login server, so it should be okay. And get connected. Yeah, so leave the other one hanging there. So it's gonna grab one eventually. So just put it on open. Yeah. And then we're gonna set up an environment. We're gonna set up a tool called Google G Drive DL.py, which allows you to download the Google Drive files directly to the Hoffman 2 server. So you can go ahead, type in these commands. Go back to my Windows. So type in the command make dir p $sign home slash bin, and let's add that path to your environment. Echo path equals $path colon $home slash bin $home slash rc. So what it does is we just created the hidden directory .bin under our home directory, and then we added this bin directory to our the local environment variable. So oh yeah, I can do that. Is it better now? Okay. So let's redo. Sorry. So mkdir -p. Just retype it real quick. Yeah, so basically what it does is it's gonna add this .bin directory, the hidden directory, into a bash rc file, which is a environment file. So once you add it, you can execute a command. Whatever the file in here can be executed globally in your console. And then let's go ahead download this script here. Actually, you gotta go to that directory. So it's gonna be home. So cd means change directory to home /bin. So I already have it downloaded here, but I'm just gonna let me just remove it real quick. So you can download it by oh yeah.
Yeah, mine's getting permission denied for some reason. Whenever I type in that echo path equals that line, I have no idea why.
Let's see. Can you actually open it up? Type in this command, -- rc. You probably see something like this.
Sorry, what was it? VI.
vi $home slash
Okay.
dot bash rc.
Okay.
Does it say you're in like read only mode, or
Where would it say that I was in read-only mode? I guess.
I mean at the bottom? If not, you're allowed to change it.
Yeah, I have no idea.
Oh, okay. So you can just type in, well, you see this export path at the bottom of the page, right?
Let's see. Under Conda. Okay. I don't see export path. No. Oh wait, yeah, I do. Yeah, I do.
Okay, so you can change to something like this. Don't copy this part though. So export path equal dollar the squiggly bracket path, and then you can type in the colon. You gotta hit the insert button before you change anything in the bash RC
Okay.
file. So you can type in the colon here at the end of the line, and you can, I guess you can do this home slash dot bin. And once you have done that, you can save the file with shift colon in your keyboard, and it's going to show up this colon sign at the very bottom. And you can type in W and Q. W means write, and Q means quit the file.
Okay.
So save and quit.
Awesome. Thank you.
Yeah. Okay, so let's get back to, let's download the file. So you can just, I guess for this one, you can just copy and paste. For rest of the commands, I prefer you guys to type in most of the command, so you guys can actually get a hands-on experience on the command, some of these commands tools. But stuff like downloading this, downloading stuff, I don't want you to guys like type in all the URLs. Oh, okay, sure. Here. And once you have downloaded, you can change it to executables. You can do that either by typing this line here. Let's just type this line. Chmod plus x dollar home bin. Actually, so what this command does is when you try to open this Python code, just give it a try. So right now it's not executable, so you can't really yeah, you probably say permission denied. So you gotta make it executable in your local environment. So you can type in chmod plus x dollar home in gdrive.dl.py. And once you have that typed in, you can probably be able to execute the file. Yeah. So if it does that, it does. If it doesn't say permission denied, then you're good to go. Please give me a thumbs up if you guys all made it through. Okay, I see two of you guys. 3. Okay, 4. All right. I think that's majority of you. Okay. And let's go back to our home directory. You can type in, you can either type in this or this to go to your home directory. So we have made the QCB workshop directory there. So go to that directory. And then, so we're gonna basically download everything to this QCB workshop directory. So what you can do is, yeah, you can type in G drive dl.py. It's everything is set up perfectly. You will see, you will be able to run the G drive DL in a different directory. And then you can copy this link. I'm just gonna provide it in the chat. Yeah, so you can copy this link, gdrive.py. Oops. Maybe the link is broken. Let me just share something else. That's weird. My process URL. Oh, that because it's on different Python. Okay. Okay. So I guess we need to change our Python version here. Yeah, let's change that. Let's change to the Anaconda one here. Okay. So you can change it by typing in this command, module load Python Conda 3. So I'm not gonna explain the stuff about Anaconda in a bit, okay? And it's gonna ask you to type in this command. So let's do that. Conda dir etc profile.d conda.sh, and let's see which Python. Yeah, so if you did everything right, you should get the Python version 3.6.5. You can exit this console by type in Control D, and let's give it a try one more time. gdrive.dl.py, and then copy in the URL. Yeah, so if you have done everything right, you should probably see this. downloading a file directly from the Google Drive. For those of you who are waiting to get a node, do you guys still waiting, or how did it go? Did you guys get any node? Bhumika and Chad? Okay. Okay, so some of you don't have command not found. So if you guys have the bash properly set up, wait. What kind of trouble?
So I tried the chmod command and it said there was no such file or directory, and I'm not sure. I don't think there's a typo.
Okay.
I don't know if I should share my screen or.
Did you see this file in your .bin directory under your home directory? No?
No, I don't think so. When I press, well, when I just list the files, all I have is the G drive dl.py.
So you have that file, right?
Yeah, but I don't have. Oh, hold on. cd. Oh wait, there's nothing in. I don't have like assembly stats G drive harvest tools.
Yeah. You don't need it for now. Yeah, this is for the later on. All you need is a G drive.py.
I don't have that. Oh,
Okay.
maybe it's in the wrong spot.
Okay. So you can re-download it with the wget command.
I should make sure I'm inside this bin folder
Yeah.
and then download it. Okay.
Yeah. And for those of you who show the bash error, do you guys also have the file in this directory? .bin?
Yeah, I have mine. The G drive dl.py is in mine, but then whenever I try to rerun the command, like with the website URL, it's still saying that the permission's denied.
No. Can you type in this? Under your .bin directory. You gotta be in your
Okay.
.bin directory.
Okay. And then try it again.
Yeah.
Okay. Oh,
Is it working now?
oh wait. No, I just got a bunch of errors.
Yeah, that's probably has to do with the Python version you're currently having. So you can type in this, load Python. Can you also type this in and retry?
Before activating. When it says before activating a Conda environment, please issue .conda dir.
Yeah. So you
Yes.
gotta type that
You got
in to.
Sorry, I think I might still not be in the right spot. When I
Okay.
type in gdrive.dl.py, it says command not found. And I'm inside the bin folder.
Okay. So in that case, you probably have missed this part, I believe. Or if you
Uh,
have typed in this part, you can. Did you type in this command?
I typed it before.
Okay. Then try to let's try to reload it with the um, and try to type in this line and try again.
Um, okay, that worked.
Okay.
What
Cool.
what was wrong?
So bash RC is your environment variable file, right? So sometimes it has to be reloaded. Like if you have relogged into your terminal, it should have been worked, I guess.
Oh,
So the source command I just gave you sort of like reloads the entire environment variables.
okay.
Okay.
Thank you.
Okay. Is anyone else also having a trouble? So everyone good now?
I'm sorry, I'm still not.
Okay.
So, yeah, I went back and redid everything, and I'm still getting like errors whenever I do the G drive with the URL on there. So like the chmod command worked, and then after that, it's not working.
But it's not permission error though, right?
No, it's not a permission error. There's just like a bunch of errors that it's saying, like in
Okay.
different lines.
Did you type in the Python? Did you change the Python version with this command?
Yeah, I
Module?
did.
Okay, can you type in the Python and see which version you're seeing right now?
Yeah. Let's make sure. Yeah, my Python is 3.6.5.
And you're still getting an error? That's weird. Can you actually try it with the Python in front? Python G drive PLPY. Yeah. And then URL.
Okay. Let me try that. Okay, it's working now. I think that's what it was.
Okay, okay, cool.
Great. Thank you.
Okay. Okay, so I guess.
I'm sorry, I'm still having trouble with downloading the file. I was trying to follow what you were telling her to do. So I did the Python to check what version I'm working with, and it's 3.6.5. But I don't know how to exit off of this little thing. It just gives me
Oh,
three.
you can press Control D at the
Control
console.
D.
Yeah.
Okay, gotcha. And then what was the next step after that?
Try to type in Python G drive PL G drive DL.py.
Okay.
and then your URL that I just
Oh, and the
gave you.
URL.
The
Okay.
Sorry, when I do that, I don't get the adapters output. I just get some errors. Like value error, 0 length field name and format.
One sec. Can you re-download it with this link again?
Re-download, you mean rerun the Python command? Or
Yeah.
so Python and then G drive dl.py and then paste the link.
Yeah.
Yeah, I still get this error.
What kind of error you got?
It might be easier if I share my screen. Would that be
Okay.
possible? Sorry, everyone. Oh, can you enable me to share?
Yeah. Go ahead.
This one.
I think it's a version issue. Can you type in Python in that screen? Yeah.
Oh, I
that's
thought
why.
I changed
Also,
it before. Oh, hold on. Can you remind me how to change?
Yeah. So module load
Okay.
Python /Anaconda 3. And type in that command too. Yeah.
Do you have to do this every time you run the Python command? Because I had
No.
I ran this one before too.
For Conda, yeah, you need to do that because try that. It's G drive DL. You have typo there.
Oh. Okay. Thank you so much.
Yeah. So once you have that downloaded, make sure to move that to QCB workshop directory, okay? Right now you're under a .bin directory, right?
Where's the QCB directory? Is that?
We just made it in the home directory, under your home directory. If there's, I mean, if you haven't typed in the command, you can just type in you can go to your home directory by cd and this. Oh wait, I'm not sharing my screen. Yeah.
Oh.
So cd and you can type in this, and once you're in the home directory, type in make dir QCB workshop.
Okay. Thanks.
And then you can move your files. I guess you can go in, and once your download is complete, you can move your files from the bin directory, which is, you can type in this command, I think. So you're gonna probably have adapters in your bin directory and have it pasted on here. PhiX, and then data. Okay.
Okay.
And then once you guys all ready, I guess we can take a break now since it's past 1 hour from the start. So we can have a 5-minute breaks, I guess. Did everyone get the node right now? Okay, that's good. Oh, really? I guess few of you still haven't got the node yet. Okay. Um, let's keep on waiting. We don't need it right now, I guess, until the later on this slide. But you guys all have downloaded the files, right? And have them set up under the QCB workshop directory. Okay, so that's good. So what you have just downloaded is the raw genome file from the cholera outbreak that happened in 2016 in South Korea. It's from one of the patient data. We had a total of 3 cholera patients in that year. So it was kind of shocking in Korea because we never had a cholera patient for at least for like 15 years before, right? And then Korean CDC sampled a seawater from this harbor, the Daege harbor, and they also found the cholera isolate from the seawater sample. So none of the patient had a traveling history uh, to outside of the Korea. So we were kind of assuming that it was a local infection, so local outbreak that has happened. So it was kind of big in the news for a while. Yeah. So that's the genome we're going to use for this workshop. And we already loaded up the Conda environment. So the Conda is the package management system. You can think of it as an app store in your mobile phone. So it provides different environment settings for different pipeline or softwares. So it gets much easier to set up certain programs and install certain programs, especially in Linux. I mean, we all suffered through like installing this gdrive.dl, right? Yeah, so once if your program is registered on the Conda package, you don't have to suffer through that. You just need to install Conda and install your software there. And the other thing is it also avoids the conflict with the different programs. Sometimes some programs require different versions of the program. For example, the Python, the versioning issue that we just had before. Yeah, so you can set up a different environment with different Python version for that case. So we won't have any conflict with the other programs. Um, so you can install Conda on your not only on the Linux environment, but also in your Mac, I think, and also in Windows as well. Yeah. So if you're not using Hoffman 2, you can install it on your local environment and use the same command line that we are using here to your on your local computers. But so this is how you install it on your local environment. But you don't have to do this for this time because we're using Hoffman 2. For Hoffman 2, you can just type in this module load python / anaconda 3. And if you have loaded the anaconda, you should be able to see something printed when you type in conda. Yeah, you should be able to see something like this. Okay. Let me just load up another screen here real quick. Okay. So with Conda, you can create and remove the Conda environment. So did everyone see this printouts when you type in Condas? Okay. So you can create your own environment. Let's make a test environment. So Conda create -n test. Yeah, so it's gonna ask you to proceed and type in yes. And once you have done that, it's gonna tell you how to activate your environment or how to deactivate. So let's try to activate it. Conda activate test. Once it's activated, you will see a little bracket telling which environment you're in right now. And you can also list up the existing environment, conda in list. Yeah, so this sort of shows you the environment you're currently have. And the star sign here shows the activated environment. And let's go ahead, deactivate it. Conda deactivate. And as I said, you can also install different Python different versions of the software. Let's say, for example, you want to install Python version 3.6, then you can type in Conda install Python 3.6. Okay, just don't type it, don't hit enter with this. We don't want to change the Python version right now. And then you can also remove the existing environment. So let's go ahead, try to remove our test environment that we just created. Test. And dash dash. Oh, that's weird. I'm not sure why it's not working right now, but this is the command to remove it. Let's skip forward next part. We did activate, deactivate. Oh yeah, so let's finally get into the stuff of a bacterial genomics. So bacterial genome has a few characteristics. So most of the bacteria has a single chromosome and consists of a few other plasmid, and they are all like circularized in shape. So the size of the bacterial genome kind of varies depending on which species you're working on. The smallest can get to like 130 kilobase pair to over 14 megabase pair. So as you can see here from the plot, the size and the number of genes, number of protein coding genes is kind of has a linear relationship with bacteria and archaea. So obviously more genome size, or bigger the genome size, more protein coding genes you might have in the organism you're working on. And they can change the genetic material through the horizontal gene transfer. So mutation can happen either during the DNA replication, which is a typical point mutation. They can absorb a free DNA element from the environment, which is called transformation. They can also obtain a genetic element from the virus, the bacterial phage, which is called transduction, or they can even share between two different organisms, which is called conjugation. And this all happens with the recombination. So it's gonna, with the horizontal gene transfer, you're gonna have a regional change within your chromosome or plasmid. So why we sequence bacterial genome? Obviously the technology, the advancement of technology, the sequencing technology has significantly increased the number of genomes in the public database. I mean, as technology advanced, the 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 price dropped significantly. So right now it only costs you like a few $100 to sequence the bacterial genome, depending on the size of the genome actually. And up until 2018, we almost had more than 50,000 bacterial genomes in the public repository. And as we have more data, it became one of the promising method to to identify species, and perform other comparative analysis. So we get to understand the bacteria better. So conventionally, people only sequenced the cultured isolates. So this is a Petri dish showing your hands, bacteria that are colonized within your hands, right? So each of this colony represent different species, I guess. So you get to isolate them, and you can spread on another Petri dish like this, and you can isolate the cultures. But the the disadvantage of this culturing method is only about 2%, less than 2% of bacteria can be culturable. So rest of the like 98% of the bacteria is still out there to be sequenced actually. But thankfully, with the advancement of shotgun metagenomics pipeline, technology, we get to obtain the bacterial genome out of the environment sample, so without any culturing steps. So from the environment samples, you can, this is for Illumina sequencing actually. So from the environment samples, you can chop up the DNA element into a fragment, so we can sequence it. And then after sequencing, we can sort of like bin the reads together that belongs to the same species and try to build up a genome out of it. But so far with the short read sequencing, it wasn't like that perfect. We could be able to get some residues of the genome, but we couldn't get full sequence. But these days we also have this long read sequencing available, stuff like PacBio or Nanopore, which allows us to rebuild the genome entirely from the shotgun sequencing. So this is still like actively advancing field. Yeah. So, but for today's workshop, we're gonna mainly focus on the cultured isolates, since this is still since the majority of the sequence performed is still under NGS with the cultured isolates. So we're gonna focus on this for today's workshop. So a little bit about Illumina sequencing. So on the each flow cell, there's this oligos attached to it, which is complementary to your DNA library you prepped, which is complementary to the adapters attached to the DNA element that you just prepped. And then they amplified this small fragment of reads, this small fragment of DNAs with the technology called bridge amplification. So depending on the cycles you perform, you can either get paired end or single end read sequenced. So what actually happens is that DNA gets synthesized with the fluorescently labeled nucleotides. So this sequencing technology is called sequencing by synthesis. So since the nucleotide is attached with the fluorescence, it's gonna emit a light in different colors based on depending on whichever base gets attached. So every time, the light emitted gets recorded, something like this, and measured, and that's how the sequence is actually performed. So obviously the light can sometimes be brighter or dimmer, depending on the signal. So that's how you measure the quality of the each base of the sequence. Little bit about paired ends. So paired end is if you have one read, so you read, you sequence the read in both ways, one with the forward end and one with the reverse end. So what it does, there is some overlapping regions between forward and the reverse with the paired end read. So if your read length is let's say your read length is like 150 base pair, with paired end, when you combine, you can actually get sequence length, I think up to like 250 or 300, which can cover some of the regions, some of the repeat regions that are in your genomes. So it's gonna, theoretically, it can improve your assembly results. So Illumina suggests for whole genome sequencing and other de novo sequencing, they all suggest to use the paired end 150, at least a paired end 150 base pair for the genome sequencing purposes. And for single end, you can apply that to the RNA sequencing relevant data analysis. So paired end is a little bit more expensive than single end. Yeah, just keep that in mind. And this is the file. The FASTQ is the file you get once you get your sequencing done. So it comprised of these 4 lines of structures, multiple lines of these 4 lines, right? So at the top, you will see your sequence ID with the @ sign at front, and in the second line, you will have your base calls. Third line is just a random placeholder, and the fourth line has a quality value for each of this base. Even though this is a character, you can convert it to a number. This is just in computer, everything is number, right? So this character can be converted to, based on their ASCII, what's called the ASCII code. You can convert them into a certain number. This number can represent certain Q values. For example, the exclamation mark here is ASCII 33, and Q value is 0. So obviously this space has really low Q values, and probability of error rate with 0 Q value is 1. It's 100%. So it's an error, obviously. Okay. But we can't really handpick all these quality values by ourselves, right? So there's obviously a program called Trimomatic, which does the quality trimming of the raw data, and it also removes the adapters from Illumina sequence. Okay, so does everyone have a node now? Okay. Okay, too. That's a bummer. Okay, so for now, I guess those of you who didn't get a node, I guess you guys can just watch what I'm typing. Now you can execute the command on the login server. So I guess you just have to wait until you get the node. Yeah. Or maybe you can install it on your Mac, on your local computer environment. Uh, I think I can help you after after this workshop. Okay. For those of you who has a node, let's try to type in this command. Yeah, it seems the node, the Hoffman 2 is really busy today. Yeah. So those of you who got the node, make sure you have this n and number in your console, just to make sure that you're under a node right now. So let's create a Conda environment. Create -n Trimomatic. It's gonna ask you to proceed. Type in yes. And after that, we're gonna activate the environment. Conda activate Trimomatic. Yeah, make sure you're on the Trimomatic environment with this Trimomatic sign up front. And let's go ahead and install Trimomatic. Conda install C Bioconda Trimomatic. So this -c Bioconda is a kind of like a repository for all the bioinformatic related tools. So you got this is it telling the Conda to look at this repository to find the Trimomatic program in the server. And hit enter. It's gonna ask you to proceed. Type in Y. Can you type in this? Yeah, so if you're working on the new terminal or console, you gotta always reload the Anaconda. So you gotta remember this command when using Hoffman 2. Okay. I'm just gonna wait a few minutes here so that everyone install their software. If you have installed your Tremomatic, can you give me a thumbs up so that I know who's complete?
Will anything bad happen if I run this on the login node?
Yeah, so the Hoffman 2 team will probably email you, warning you.
Oh,
You're not
okay.
supposed to run any programs on login node. Yeah.
Okay.
But I guess after this workshop, I can help you to set up the local environment so you can try it on.
Okay. Does it run? You said it runs for 6 hours?
No, the typical, I think for the assembly, it takes like 1 to 2 hours
Okay.
to complete. But I just reserved the 6 hours just to make sure we get extra time. Yeah.
Okay. Thank you.
Yeah, I guess for tomorrow, before you joining this workshop, you can go ahead and try to grab a node. Probably try it like before a couple hours. Yeah, maybe give it a generous time, like 6 to 8 hours. Yeah, so make sure you guys have the node before you join the workshop. Okay. Okay, so I saw 2 thumbs up so far. Okay. I see Sarah's ready. Okay. Yeah, so it takes some time depending on the which type of node you got. I mean, some nodes are really old, and so they're sometimes somewhat slow compared to other nodes. Is everyone else also waiting? Yeah, I guess I'm just gonna proceed this time. You guys, once you have it installed, you can just follow through what I have shown, okay? Once you have it installed, you'll be able to type in Trimomatic, and you will see some printouts if it's properly installed. And for this time, you can change your directory to raw data. So cd raw data, which consists of the cholerae raw data file here. So you can start typing in, fill in these blanks here. Trimomatic, PE for the paired end, and we gotta put in two raw read data, which is this one. The second one is Vibrio, this one. And then you gotta type in the output file. So let's type in Vibrio 1. Actually, let's type in QCV VC QC dot paired fq dot gz, and then we gonna put in the unpaired output file. So sometimes this paired end don't have a match with the complementary pairs. So that's where this unpaired file, this is what this unpaired file stands for. Okay. So QCV underbar VC QC dot unpaired fq dot gz, and for the second read output, we can. Oh, actually, we have to. So this is the first read. So we're gonna designate with number 1 here. Same for this. And QCV underbar VC dot read 2 QC paired fastq dot gz, and then QCB underbar VC dot 2 QC unpaired FQZZ. Yeah, and then we're gonna type in the Illumina adapter information, which is provided under the adapter folder. So we're just gonna type in Illumina clip. This is all in capital, so keep that in mind. Colon // which indicates the upper directory. So right now, if you've done everything correctly, you are under the raw data directory, right? So one above that, we have adapters folder. So we're gonna type in adapters. So this typical Illumina data used the TrueSig3 library. So you're gonna use that. So TrueSig3 PE -2.fa. And then make sure there's no space between the parameters with the Illumina clip. And colon 2, colon 30, colon 10. I'm gonna explain what this means in afterwards, okay? Colon 2, keep both reads, and for leading, remember everything is in capital. 3, trailing, colon 3, space, mainland, colon 36. And for threads, we only reserved like one thread for this node. So we're gonna type in 1. Yeah. And then you can hit enter. And it's gonna, if you type in everything correctly, it's gonna start trimming out the raw data. I'm just gonna give you the command I just typed in under the chat. Yeah, so a little bit about the Illumina clip. So it is structured in this way. So it comes with a fast adapter first, which is the adapter file we provided under the TrueSeq 3pe.fa, and seed mismatches. This one means that if there is like more mismatch, if there is a mismatch in adapter sequence, let's say if there is, we gave a 2, right? If there is a more than 2 mismatches with the adapter sequence, then we're gonna discard that read, right? So let's say if if our sequence has something like GG, right? If if our adapter was something like this, and we consider that the adapter is contaminated, and we discard that read, okay? And for the palindrome clip threshold, so for paired end, when you merge 2 pairs together, you get this like palindrome like shape of sequences, right? So once you align them together, this is like an alignment score threshold. If alignment score is less than 30, it's gonna drop off the paired end. And for simple clip threshold, it's just a alignment of alignment score of adapter sequence only. Yeah, so if it's less than 10, alignment score is less than 10, it also gonna remove those reads. Okay. And keep both reads. This one is kind of important for the further downstream analysis for the genome assembly. So once you remove the adapter sequence, let's say this adapter sequence got removed after the trimming, and then let's say all the overlapping reads are same, right? Without any difference. If those two are redundant, Trimomatic by default is gonna remove the read to data just to save some, I guess, save some space because this is redundant data. But for further genome assembly pipeline, we do need both of this information, so we want to keep it, right? And then two here is this number of bases of adapter that you're allowed to keep. Yeah, so and then for leading, so for the forward end part, so I've told you about the FASTQ scores, right? So from the forward end, the leading, the score threshold value is 3. So if Q value is less than 3, it's gonna remove that base. Same for the trailing. This is the threshold for the reverse read. It's also gonna remove those bases. So what happens is with Illumina data, the quality kind of gets dropped as you go to the end of the reads. So it's gonna trim most of the like end part, but you still get some regions left to get overlapped with the second reverse reads. Okay. And if you trimmed too much, let's say, and then your read length gets dropped into like under 36, it also gonna drop the entire reads. So that's what this program is doing right now. I think it's gonna take roughly about 10 minutes to finish. So maybe a few more minutes. Yeah, I guess this is a good time to take a break for now. Let's take a 5-minute break. Hopefully this gets finished by then. Okay. Is there any questions so far? I think you're under a wrong directory there. Are you under the raw data directory? Yeah, you have to be under the raw data directory where the file is. Here. So in your directory, you should have these 2 files that we downloaded from the Google Drive. Okay. Okay, let's take 5-minute break. Yeah, so as you can see, the streamomatic is done. So a lot of reads are pretty much in good shape. So 99.79% survived actually. And I mean, this is already kind of quality filtered data. Yeah, so that's why the surviving rate is so high. But with the real raw data, I guess that the surviving rate will be a little bit lower. So another thing I'm going to talk about is the PhiX genome. So the PhiX genome is a bacterial phage genome, the virus, which has like really short genome size, which is about like, I think 5,000 base pair long. So what people do is they artificially insert the PhiX genome into their library so they can use it to calibrate the sequencing platform. So if they can retrieve the PhiX genome from the sequence data, which means the sequence have been done okay-ish. But oftentimes people like forget to remove this PhiX genome, artificially inserted PhiX genome. So what people do is they usually do the quality filtering with the Tremomatic, and then they forgot about PhiX genome. So there is a residue of the PhiX genomes in your genomes. So apparently, there's a study about this. So in 2015, Spratim et al. Have conducted this large-scale evaluation of the bacterial genome sequence. Turns out they looked up for 18,000 public genomes available out there. So they found out about thousand of them contains contamination with the PhiX genome. So when you are dealing it, just add when you are quality filtering, uh, keep in mind to add in these extra steps to remove the PhiX genome. So we're gonna to do this, we're gonna use the mapping program called Bowtie2. Let's go ahead and install that. So we are done with the Tremomatic. So I'm gonna deactivate it with Conda deactivate. Oh, sorry. And then you're gonna install Conda. You're gonna create a Conda environment for Bowtie2. Conda create and Bowtie2. You can run this command on the separate terminal or console, I think, if your Tremomatic is still running. And little tip here, you don't have to type in two separate lines. You can merge these two lines together. So when you're creating, you can conda create -n Bowtie2, and then you can also type in this part at the end to make it one line command. Bio conda Bowtie2, and then you can you can type in --yes to say yes to every prompt that is asked, and you can type enter. Sorry, it takes a while to install. Okay. And then after installation, you can activate conda activate Bowtie2. Yeah, you see that it's activated with the little bow tie sign up front. And then you can now change your directory to PhiX directory here. So cd PhiX. So there's a PhiX genome that I just downloaded from the NCBI database. So we can, since you're gonna use it as a reference, we gotta index it with the bowtie command, something like this: bowtie2 build, and then nc and the file name, and then the prefix. I'm just gonna type in PhiX. Yeah, so if you have done everything correct, you will have this printouts, and then the index file being generated, right? Actually, let's copy the quality filtered data from the Trimatic folder. So we have the QCB files here that we just quality filtered with the Trimatic. One thing about unpaired read, I think we can merge them together since they're unpaired. There's no meaning to have a separate files for these two. Let's put them together with the cat command. Cat, it means just concatenate two files. QCB VC1 paired with QCB VC2 unpaired, and greater sign, and then let's name it QCB VC, just QC unpaired. Yeah, so if you type that in, it's gonna create this merged unpaired file. So we're gonna use that as well. So I guess we can delete the files we don't need. Let's delete the raw data file. Okay, so you can delete the file with the remove command, rm the file name and -f. And then let's remove the unpaired reads. Okay, remove QCB. All right, so now we have three files that we need for the bowtie mapping. So let's go back to PhiX directory, and actually let's go up to the QCB workshop directory. So let's type in the command bowtie2 -x PhiX directory. So X stands for the reference. So our reference is in the PhiX folder. So we're just gonna put the directory there, and the reference file, which is indexed based on this prefix PhiX, and -1 for forward read, which is under our raw data, right? So raw data QCB VC .1 QC. So we're gonna use the paired data here. So we're gonna use the forward paired data, and 2 raw data QCB VC2 for the reverse. And for the unpaired data, unpaired data can be also useful when it comes to genome assembly. So it can work as a bridge between the gaps. So we're gonna incorporate that as well. So we're gonna use raw data VC, no, QCB underbar VC unpaired. .qc unpaired. And we're gonna save the files that is not mapped. So we're gonna separate the reads that are mapped to the PhiX genome and that are not mapped to the PhiX genome. And the reads that are not mapped to the PhiX genome can be saved with the --on conc. And we just can type in the file prefix with PhiX free with the reads that are aligned to the PhiX genome. We're just gonna save them under different file names with the prefix PhiX. And S is just a SAM file generated with the mapping program. This is something we don't need for now, so we're just gonna put it in a temp. And -p stands for the CPU power we're gonna use. Since our node only has like one CPU power, we're just gonna put in the 1. And we're gonna hit enter. Yeah, and then it's gonna do some mapping there, removing the PhiX free genome raw data. Okay, so this is roughly gonna take like 3 minutes. So is everyone following okay so far? Or I guess some of you are still waiting for node or Trigonometric. I guess the silence is good, right? Uh, so it was this is the cat command I was using. So we were combining these two files, qcbvc.1 with qcbvc.2. So we are combining these two into this file. This is what it means. Are you asking what comes after PhiX removal, Maura? And then we're gonna assemble the genome. We're gonna use those PhiX free raw data and put them into an assembler. Um, so I guess Jonathan, I guess I can help you out after the workshop. I'm not exactly sure what's going on with your environment right now. Okay. So I guess we have, so bow tie is done. So let's see if we have a right. Yeah. So basically we have two PhiX free genomes. I mean the read files. Yeah. So we're gonna feed them into. I mean, actually, yeah. So apparently, this data has already a PhiX removed. So that's why the alignment, there is a 0% aligned with the reference. So you can actually just go ahead, use the, I mean, you can use this file, I guess. Yeah. So next thing, we're gonna actually perform the assembly pipeline. So the program we're going to use is a Spades, which is a very common assembler for the bacterial genome datasets. It supports different sequencing platforms, Ion Torrent, Illumina Sequence, PacBio. Oh, there's two Ion Torrents. Okay, PacBio, and possibly Oxford Nanopore, I think. So what it does is like each read is gonna separate each read into different size of the fragments. So for this case, they're gonna use the length, substring of length 3, right? So this is called the Kamer. So this is a dreamer, right? So since we separate each string into 3 characters, character string. So we're gonna have a list of this uh these Kamers, and we're gonna generate a graph out of it, right? And we can apparently generate a directed graph. We know from the read information that TCG comes after ATC, followed by CGC, and then GCG, GCA, CAA. So this unique Kamers can become a node for this graph, and edge representing the direction of this the direction of this Kamers based on the read they are from, right? So let's say if you don't, let's say this, you don't know the entire sequence and you're just given with this graph, do you think you'll be able to like reconstruct the entire sequence just by looking at this graph? The answer is yes. You can just follow along this graph, right? So starting from ATC, it comes with the G and then C, and then if you there's two paths here, right? You can just take this path in the first place or this path. But if you take this path, there's no way going back to this node. So you wanna take this path first. This is the obvious choice, right? And so GCG, then CGC, then GCA and CAA. So you can reconstruct your entire string, the read, based on this graph. So they're gonna draw a De Bruijn. This is called the De Bruijn graph. So they're gonna draw this network structure with all the reads, and then they're gonna measure the overlaps of these k-mers. And then with that graph, they're gonna finalize the assembly. This k-mer size can be varied and can produce different results based on the different size of the k-mers, which can be varied depending on the size of the genome that you're working on. So Spades suggest to try out like different sets of k-mers. The more k-mers you put in, the more time it's gonna take. Sorry, one. Yeah, sorry about that. So more, I guess you can try out very different size of the k-mers for Spades. But actually there's this wrapper program called Unicycler, which also incorporates the Spades, which basically runs the Spades, but it's gonna use your sequence information to find the optimal the k-mer size for your assembly. So we're gonna use Unicycler. Once you, I mean, if you install Unicycler, it's gonna automatically install Spades as well. So let's go ahead and install it. And Conda deactivate. Let's go ahead, type in this command, conda create -n unicycler c bioconda unicycler, and then we're gonna install other program called SamTools. I'm typing this in because we need a specific versions of it. Yeah, 1.11. So SamTools equal 1.11. Yes. Type that in. Let's wait a few moments here. Okay, so Once it's installed, you can activate it. Cycler. Okay. And you can type in this command to assemble your genome. So unicycler -1 for the forward read, PhiX 31-2, PhiX 32, S for unpaired read, but since we sort of merge them together with the PhiX result, I guess we can skip the unpaired FASTQ file. So -O for the output directory, let's name it unicycler out. And for thread, we're gonna use 1, and we're gonna put no correct at the end. So this no you don't need to start Conda init. Yeah, but every time you reopen the console or get a new node, you can always reload your Conda with module load command. Yeah, so no correct at the end here means like space does have like their own quality filtering scheme. But I don't think they removed the adapter sequences. So that's why we do the Trimomatic ahead before we actually running the unicycler. I mean the spades pipeline. So since our data is already quality filters, we're gonna use no correct options. So the space won't run their own quality filtering steps. Okay, so we're just gonna type that in. Oh, there's typo. And it's gonna start running. It's gonna take about an hour or two to finish up the assembly. So I guess this is it for today's workshop. For tomorrow's for tomorrow, I think I hope you guys have this assembly files ready. So keep them on while it's running, okay, until it finishes. And yeah, that's it for today. And those of you who have like trouble with the technical issues, you can stay after and I can maybe help you out, okay?